Hello, welcome to 13 Universal. We are on again solving A-level CIA biology. I want to say that all the questions we're solving here came from CIA past questions I'm asking, okay? And this is schedule. We are looking at all topics paper 4, A2 syllabus series 7. So watch the other series 1 to 6, okay? So get set. Let's get back to our journey of solving past questions, right? So let's apply some concept, guys. Number one, a fig 1.1 is a drawing of longitudinal section of a human kidney. All right, that's a human kidney, guys. Fig 1.1. So use the letters A, B, C, and D in fig 1.1 to complete table 1.1. Each letter may be used once, more than once, or not at all. For each description, list all letters that are correct. Okay, let's look at it, guys. Okay, pause the video and answer this question. Okay, pause it. Remember that how this works, you must solve the question by yourself, and then you watch the video to see how we solve it. Okay, welcome back. This is location of loops of Henle. Loops of Henle are found, find it more in medulla. Medulla is C. Okay, medulla is C. So you find it in C. Okay, though you can find part of them, all right, in cortex, but those of Henle are found more in C, where there is hypertonic tissue fluid. All right. All right. Location of Bowman's capsule is found in the cortex. A is cortex. It's found in A. Location of glomeruli. Glomeruli is also found in cortex. Okay. A. Contains urine at final concentration. That urine at final concentration is found in collecting duct, which can be found in pelvis. Pelvis is B. Okay. D was very important. D is the ureter. All right. So D. All right, and B. So that's the answer here, guys. Okay. So, all right, let's go on. Now let's go to the next question. The volume and water potential of the urine produced by kidney vary according to the water potential of the blood. This is a result of osmoregulation. Describe the role of aquaporins in osmoregulation. So pause the video and describe. Give us the main, the sequence of events that occurs, all right, in osmoregulation by aquaporin. What is their role? Okay, welcome back. Now, the first thing you have to know is that aquaporin, you have to say that aquaporins are water channels, right? So aquaporins are found, you must say where are found in collecting duct, which is where this happens. Also found in distal convoluted tubule. So aquaporins, all right, increases permeability of collecting duct, the membrane of collecting duct. So increase in the number of aquaporins increases the permeability of the membrane of collecting duct, permeability to what? To water. So water is able to move from the glomerular filtrate in the lumen of collecting ducts into tissue fluid or into the blood by osmosis. Look at it here. And the aquaporins are water channels. This is very important. Aquaporins increase cell surface permeability. Yeah, that's what they increase permeability to water because they are water channels. They allow water to pass. Okay, so water channels, so water can pass through them. So and they're found in collecting duct or distal convoluted tube. So they allow water to be reabsorbed or move into tissue fluid or blood. Okay, 3 marks. So describe the role of the brain in osmoregulation when water potential of the blood increases above the set point. Pause the video and try it, okay? Now, when the water potential of the blood increases above the set point, what happens is that the osmoreceptor, which is found in the hypothalamus of the brain, the osmoreceptors dictate the change, the increase in water potential above the set point. It dictates it, and then it sends less impulses to the posterior pituitary gland to reduce the secretion of ADH into the blood. Now the posterior pituitary gland actually secretes ADH into the blood. So once the blood water potential is higher than the set point is above the set point, so less ADH needs to be produced. Okay, so that means that osmoreceptors will send signals, impulses to posterior pituitary gland to reduce the secretion of ADH. Look at it. So first of all, it's dictated by the osmoreceptors. They are all found in the brain, hypothalamus. This is found in the hypothalamus. So also osmoreceptors then send fewer impulses to posterior pituitary gland. This is PI, this typo. Okay, so less ADH release, okay, into the blood. All right, 3 marks. Now look at this question. I want you to read this question carefully. You know, a lot of background information. You must be able to remember it. So let's go, guys. Interferon alpha IFN can be produced as a recombinant human protein to treat some types of cancer. The gene IFNA2, this is the gene, codes for this protein. This gene codes for this protein, and this protein is used to treat cancer. Is that okay? So you have to code it. Now, one method of producing recombinant interferon uses genetically engineered Escherichia coli bacteria that contains recombinant plasmid. So this has recombinant plasmid. A recombinant plasmid has two, of course, DNA from 2 different sources. Each recombinant plasmid contains, look at what is found, the gene IFNA2, 3 regularly sequences of lac operon, promoter, operator, and lac-I, a gene for antibiotic resistance, AMP3. If you remember, this promoter switches on transcription operator. So this is the start point of transcription. This ons it. It switches it on. So this regulates it. Okay. A gene of antibiotic resistance. This, if you remember this topic, this is a marker, gene marker, right? So each of these genes for I, that's the regulatory gene, and this gene AMPR, okay, ampicillin resistant gene, contains its own promoter so they can be expressed. As a result, these genes are always expressed, so they have the promoter, they can always be expressed in E. coli bacteria that contains this recombinant plasmid. Guys, did you get this? You must get this G before you continue, and remember it. You must remember it, okay? If you did not remember it, go back and read it again. Pause the video and read it again. Now, let's go on. Fig 2.1 is a diagram of the recombinant plasmid. The promoter region of lac-I gene and AMPR gene are not shown. So look at it, guys. Very cute. This is promoter operator for this. All right, the promoter for this gene and this gene are not shown. Okay. All right. Look at the question. Now, another background information. The start of transcription of gene IFN2 by E. coli with the recombinant plasmid shown in Fig 2.1 needs to be controlled to obtain an optimum yield of IFN alpha. So this protein, we need to control this to obtain maximum yield, okay, optimum yield of this. Scientists investigated the effect of 2 inducers of transcription. These are the molecules that cause transcription to occur on the production of recombinant IFN alpha. So lactose, which is converted to allolactose. So don't forget this. Lactose, which is converted to what? Allolactose in E. coli. IPTG, which is a synthetic molecule with a very similar structure to allolactose. So they have similar structure. That means they bind to the same site. IPTG cannot be broken down. So please don't forget this information. You're going to need them. Cannot be broken down by E. coli. The scientists grew 3 cultures of E. coli containing recombinant plasmid in the same growth medium. The growth medium contained glucose, amino acid, essential vitamins, and minerals. The growth medium did not contain lactose. After 4 hours, this is very important. After 4 hours, either lactose or IPTG at the same concentration was added to 2 of the cultures of E. coli. As a control, the 3rd culture of E. coli was grown without adding lactose or IPTG. So the 3rd culture didn't have just as serving as baseline, right, or control for comparison. Now, the concentration of recombinant IFN alpha in the cultures was measured at different times over a period of 12 hours. Okay, yeah, okay, 28 hours, sorry. The result are shown in fig 2.2. That's the result, guys. Now you need to pay attention to this. So this is time, all right? So 28 hours. Okay, this is 0 to 28, all right? Now this is concentration of this protein. So like I told you, anytime you're given a graph, the first thing you have to check the general trend and also check the value of one box. From here to there is 10 units, 10 boxes, so 10 divided by 10 is 1 unit. From here to here is 10 boxes, 100 units, so 100 divided by 10 is 10 units. So one box is 10, while one box is 1. Is that okay? So you have to look at general trend. So this is culture to which IPTG added, culture to which lactose added. Okay, control. So control, you can see that in control there was no, okay, the concentration of this IFN alpha remained, all right, remained constant, while that of lactose increased and then decreased. But if you look at IPTG, all right, there's a steep increase from here. This is, if you count the boxes, that's 1, 2, 3, about, that's 4 hours increases to peak. Okay, if you bring it down, that's about 8 hours. So from 4 hours to 8 hours, there's a steep increase. So it peaks as 8 and then it decreases steeply, less steeply, from 8 hours, okay, of course, to this point, which is about 15 hours, okay, and then decreases less steeply. Okay, so overall IPTG, all right, increases more than them, all right, causes an increase in the concentration of this protein IF and alpha. So the question I want to be thinking about, why does it produce more higher concentration, okay, increase in the protein more than the lactose and control? They'll ask you, and why does it decrease? Why does this protein decrease? Okay, probably they're being broken down or thereabout. Okay, all right, so these are the things you have to have in mind. Let's go on, guys. And of course you can see, all right, this is, the peak is 8 here. Okay, all right, yeah, so from 8 down it decreases less steadily. Okay, see it here. Now the first question there says, the regulatory sequence of lac operon contained in the recombinant plasmid are involved in the control of transcription of gene IFNA2. Explain the role of this gene LAC-I in the control of transcription of the IFN2 gene between 0 hours and 4 hours. Can you see 0 hours and 4 hours? This LAC-I is working because the gene is being inhibited, is not being expressed. How do I know? The protein concentration remain the same. Okay, so then I say, explain the role of gene this lac regulatory gene LAC-I in the control of transcription. Now I want you to try this question by yourself. Okay, what is the role? Explain the role. Okay, now the role of this gene is that the gene is a regulatory gene. So what does that mean? It codes for a repressor protein. Okay, that repressor protein prevents transcription from occurring. How? That repressor protein binds to the operator and prevent RNA polymerase from binding to the promoter. Please don't forget that. Okay, so LAC-I is a regulatory gene. It codes for a protein, repressor protein, which prevents transcription. How does it prevent transcription? It binds to the operator and prevents RNA polymerase from binding to the operator. Okay, so transcription of that gene, the gene for this protein, that is this IFNA2, does not occur. So look at it. So this is not regular. It's a regulatory gene, codes for repressor protein. All right, so prevent transcription. So RNA polymerase, okay, so RNA polymerase, so it prevents RNA polymerase from binding to promoter. All right, that's how they work. With reference to Fig 2.2, describe the changes in the concentration of recombinant this in the culture containing IPTG from when IPTG was added at 4 hours to the end of the experiment. Now we have looked at that, but I want you to go back to that diagram. Go back to that diagram and answer this question, okay? To that graph. But we have looked at it that from 4 hours to 8 hours, there's a steep increase. It increases steeply, all right, at the beginning, all right, okay, and then peaks at 8 hours. Then it now decreases, right, less steeply. Okay, look at it. Concentration of produce increases steeply. So when this is added, so reaches the peak at 8 hours and then decreases less steeply. Final concentration. Now final concentration is greater than the starting point, so you have to pay attention to the graph, to the number. Then you have to put data too. Suggest one reason for the differences between the concentration of recombinant IFN alpha in the culture at 8 hours in the presence of lactose and concentration of recombinant IFN alpha in the culture at 8 hours in the presence of IPTG. All right, so pause the video and try to answer this question. Okay, one reason for the differences, you know that the concentration of that protein IFN alpha, right, for in the presence of IPTG is far greater than, higher than the concentration of the same protein in the presence of lactose. Why? Okay, why? Because remember that lactose is broken down. We are told that lactose is converted to allolactose. So because it's broken down, remember that two of them are inducers. So they bind to the repressor protein, okay, at the allosteric site, and then prevent it from binding to the operator, so that RNA polymerase can bind to the promoter, okay, and then the gene can be transcribed or transcription can occur. But you see, because lactose is converted to allolactose, so the concentration of lactose is low, okay, so fewer lactose bind to the repressor, so less repressor is actually inhibited. But you see, IPTG is a synthetic molecule, so it continues to increase, so it has high concentration, so it binds more to the repressor, all right, and then inhibit the repressor more, thereby switching on the gene for IFN alpha more. Okay, so more of this is there, and because we have more concentration of this, because it's not broken down, so we're going to have all right, more affinity for this, and more of that protein will be synthesized. Look at the answer here. So they say 1, IPG is higher, okay, lactose is lower, because lactose has to be converted to allolactose. Lactose is broken down, so needs to be continually taken up, so it has to be taken up continually. It's broken down. IPTG is not broken down, so continually binds, it's always binding to repressor. IPTG will be at higher concentration than allolactose, okay, or IPTG has higher affinity for repressor protein than allolactose. That could be a reason. Lactose used up, concentration decreases, so concentration of lactose is always decreasing, is always converted to allolactose. Okay, so let's go to the next question. So more IPTG enters. That's it, guys. So just one reason for the change in the concentration of recombinant IF1 alpha in the culture containing IPTG from 12 hours to 16 hours. Now go back to that, pause this video, go back to that graph and suggest, give a valid reason. There are many reasons. Why does it decrease? Okay, in IPTG. Now IPTG is not broken down, so why does it decrease? Why does IF alpha decreases? It decreases because most likely it is broken down in the blood. Okay, it is broken down, is a protein. Okay, so it's transferable and it's broken down. Now look at the next question. Gene AMPR in the plasmid shown in Fig 2.1 codes for a protein that provides resistance to antibiotic ampicillin. So this codes for antibiotic resistance to ampicillin. Okay, all right, so just how ampicillin allows genetically engineered E. coli containing recombinant plasmid to identify. Remember, I said it at the beginning that this is a gene marker. Yes, an identification of genetically modified bacteria. So pause the video. How can we identify, all right, how does it help us to identify our GM E. coli, right? Try it, guys. Try it. Okay. Now remember that if this gene codes for antibiotic resistance, okay, for ampicillin. So how you can identify it is that when this genetically engineer is grown in medium that contains antibiotic, right, ampicillin, it survives. So the one that has this gene will survive, will grow. The ones that don't have it, all right. So E. coli that have taken up the plasmid will grow, survive in the presence of ampicillin. Those who don't, the E. coli that don't have it will die. Okay. C. Bacteria can evolve antibiotic resistance through natural process. I told you they're always asking this question. Okay, they're always asking this question, guys. Outline how bacteria can evolve to become resistant to antibiotics. Okay, this is natural selection. The first thing you have to say, pause this video and write the answer. If you have been watching our videos, you've seen that we have solved this question several times. So they repeat it. This is one of the questions they repeat, okay, all the time, because it's very important, it's very critical, you know. Pause the video and answer it. So the first thing you have to say is that, of course, the mutation, all right, so mutation occurs, random mutation occurs. Remember that mutation is what introduces a new allele. Okay, previously the bacteria did not have any antibiotic resistance. Okay, so how did it show up? So mutation occurs, random mutation occurs. So when the random mutation occurs, all right, that causes a change in selection pressure. So it introduces directional selection. Okay, so remember that when it occurs, when mutation occurs, it creates a new gene, right? All right, that new gene now, all right, gives the bacteria advantage in the presence of antibiotics. Okay, so antibiotic is introducing antibiotic is like introducing a new selection pressure. That's directional. So directional selection is when you introduce a new selection pressure, change in selection pressure. So now you introduce an antibiotic, all right, that will cause directional selection. Okay, so bacteria that have this gene, all right, will survive and pass on the gene, all right, horizontally to other bacteria through the plasmid, all right, or they pass it on vertically to their daughter cells when they divide asexually. Okay, and then what happens is that their allele frequency increases, right, and that leads to evolution. Look at it. Random mutation, natural selection. Okay, this is natural selection. We're talking about directional selection. Antibiotic act as a selection pressure. Bacteria with mutation have selective advantage to survive, reproduce. Bacteria with gene that codes for antibiotic resistance have selective advantage to survive and reproduce. They pass on this mutation or gene by binary fission and sexual reproduction, vertical transmission, or they can pass it, all right, by transduction, transformation, conjugation horizontally to other bacteria. So that increases the chance of resistance if people do not, okay, if people do not of course do not finish course of antibiotic over, okay, antibiotics, or overuse antibiotics. Some antibiotics may act as a mutagen too. So please note this question comes out all the time. All right, let's go to the next question. Salmon can be genetically modified, GM, to produce, or genetically modified to produce increased quantities of growth hormone, which is a protein. GM salmon modified in this way have a faster growth rate and reach their maximum body mass at a younger age than non-GM salmon. Within any population, a population of salmon, there is variation in body mass. This is an example of continuous variation. Explain what is meant by continuous variation and how it can be caused. Exactly, this is it. This question comes out all the time. Pause the video and try it. What is meant by continuous variation and how it can be caused? Two questions in one. Now, what is meant is that it is the type of variation where, all right, the phenotypes has intermediates. There are intermediates, all right? Okay, so there is no distinct or category groups in the phenotypes, all right? And is usually quantitative. The phenotype is quantitative, so it is, all right, it shows normal distribution, right? Okay, most of the phenotypes are found within the middle range, all right? So of course we have mentioned that it is caused by more than one gene, and that's what brings us to how it can be caused. It can be caused, all right, due to the additive effect of many genes, okay, interacting together can cause this variation. And it can also be caused by environment. So it is influenced by environment. So look at it in order. Any2 phenotype has a range of value. Please memorize this. And I don't mean cram it, but in biology you have to remember this. Absolute. Okay. Of course, points. Phenotypes not in groups, so they're not in groups. All right. Or phenotypes are quantitative, normal distribution. So this is how they are caused. Polygenic, controlled by many genes. Different genes alleles have additive effects. Environmental factor can actually affect that. All right, like diet. Okay, so 3 marks. Scientists investigated whether injection of very young non-GM salmon with recombinant growth hormone could cause an increase in the growth rate of salmon. The scientists used 2 groups of trying to inject it, okay, non-GM salmon, a control group of increase in the growth rate of the salmon, all right, an experimental group of salmon that were injected with 1 microgram of recombinant growth hormone at the start of the experiment and once a week. So for the next 6 weeks, that's a lot of work, man. The mean body mass of the salmon in the 2 groups at the start of the experiment was the same. After 6 weeks, the body mass of every salmon was measured again. So they were injecting these 2 groups, okay? So this is number of, okay, no injection with recombinant growth. This is injected with recombinant growth. So you see, now a student decided to use t-test. The t-test should be performed on the result shown. Now, why should t-test be used here? Okay, all right. Remember that to use t-test, there must be normal distribution, right? And there must be quantitative continuous value. And don't forget that to use t-test, all right, we should have more than 10, okay, number. Okay, this is 28, this meets it, okay, but there's a problem here. The standard deviation here should be similar, but they're not similar. Okay, all right, let's go on. All right, that's the table. So calculate the value of t, that is t-cal, for the results shown in table 3.1 using the formula for t-test. Okay, yeah, this is the formula for t-test. It will be given to you. This is squared. All right, look at it. All right, just give your answer to 2 decimal places. This is the mean, the first mean. This is second mean here. First mean minus second mean. Okay, absolute sign. The standard deviation for the first one is this. Standard deviation for the second one is this. So divide them by the number. The number here, number is 28. Number here is 27. Okay, so work it out. So see the answer here. See, yeah. So this is the denominator. Now this is the numerator. The first one, okay, minus, or anyhow, you get 1.7. This is the denominator, okay. This squared over 28. You can see that. So what we have here, t calculated value is 7.56. Just basic mathematical calculation. 7.56. All right. Now the question says the critical value at probability of 0.5 for this data is 2.01. Guys, don't forget that 2.01 is t tabulated. So you remember we always say, okay, if t calculated is greater than t tabulated, yeah, t tabulated, reject the null hypothesis. All right, the T calculated is 7.06 there. Okay, 7.06. Yeah, thereabout. Check it again. And then T cal is this one. They calculated from the table. Our probability, critical probability of 0.5 is 2 point. So T tab, sorry, is 0.1. So let's go on. Okay. So the students used the result in table 3.1 and the test to conclude that the injection of recombinant growth hormone caused an increase in the growth rate of the non-GM salmon. Okay, comment on the extent to which the conclusion made by the students can be supported. Right. So if you look at, guys, look at this. The t-test calculated is greater than the t-test tabulated. That's 7.06 greater than 2.01. And anytime this happens, you simply reject the null hypothesis, right? That means there is a significant difference between, all right, that's what the result tells us here, between the two groups. Okay, so null hypothesis is rejected. All right, however, t-test was used, right, once. There was no repetition, so it may not reject it. Okay, because you have to state, data that support and the one does not support it. Okay, all right, if you check, you see that the t-test should be used when standard deviation is similar, but standard deviation is not similar. Okay, so like I told you, the first thing to say is that this is greater than that critical value, so the difference between the two groups is significant. Yes, so because this says we should reject, if this is greater than this, so reject the null hypothesis. Null hypothesis says that it is not significant, so reject it means that the difference between them is significant, so it's not due to chance. This is also used for chi-square, the same interpretation, right? All right, so or null hypothesis is rejected, any two, and of course if you check there's overlap, that overlap, all right, contradicts, all right, but we're just trying to discuss the result. There's overlap. Okay, if you go to the table and check, add the standard deviation to each mean, add and minus, do it, you see that there's overlap there. All right, so it's overlap there. Correlation does not prove causation, so just correlation. So only a single investigation, so small sample size was used, which could have affected the result. Reference to this at 5% probability, so may not actually be different. Okay, so these are the reasons. T-test may not be valid due to unequal. So we don't use t-test. It may not be valid because standard deviations are different. They should be similar here. All right, let's look at the next question. So just one advantage other than cost of farming GM salmon that produce increased quantities of growth hormone instead of farming non-GM salmon that you have to inject. All right, so if you are going to farm, anyway, pause the video and try it like we do. Pause it. But the advantage of using this GM is that you don't have to inject the salmon every day, so it's less stressful. Okay, and because you don't have to inject it, there's less risk to infection. Okay, all right, let's look at it. So less labor intensive, either less stressful, risk of infection, either the growth hormone is continuously produced, GM salmon grow faster than injecting this. Okay, all right, thank you for staying through. Please watch this video over and over and always try to solve it with us. All right, see you next episode. Bye. This is Shedro.